Okay, so while you guys are doing your activity 2, or doing it along the way when I'm explaining this video, hopefully it can be a guide of you to where to put in all those 3 elements, because one of them is the polylinker. So polylinker may be added at either ends, okay, of your transgene, either at the 5' upstream or 3' end, the downstream. But make sure that your polylinker, which is also known as an adapter, contain sequences of several restriction enzyme sites. So this is why, okay, because for some of you, maybe you can guess, this is to facilitate with the cutting to be ligated to your vector of interest. Okay? So transgene could then be inserted into plasmid vector and clone once you have put in the polylinker. And the transformed bacteria could then be identified via survival selection culture techniques. This is done by tissue culture. And the transgene construct is purified, and before introduction into the embryo, of course, you have to test it for expression, because you don't want to waste your time and effort just not to have the gene express it on. So you have to make sure that it could be expressed before you proceed with the transfer into the surrogate mother. So even though it may seem easy for DNA construction and all the workflow, but in real life, it actually takes several months up until years to construct transgene. And if you remember what I told you about gene construct in transgene plants, sometimes, well, it involves luck. So this is the overall chart, okay, of the transgene construction, okay, whereby it is proceeded with cloning and evaluation of in vitro to make sure that your gene expresses, followed by transfer of the gene into the eggs via any one of the method. This is by microinjection, followed by transfer to the surrogate mother and analysis of the founder animals or the transgenic animal. So let's have a look at the gene transfer methods. So we have several gene transfer methods, and I'm going to be discussing about 4. So let's have a look. Now, we have blastomia or embryo aggregation. We have teratocarcinoma cell transfer, so it involves cancer cell. We have retroviral infection. We have microinjection. We have electrofusion, similar to electroporation, nuclear transplantation, embryonic stem cell transfer, sperm-mediated transfer. I guess I'm sure you can guess, okay, based on the name. And the last one would be particle bombardment, ballistic, similar to plant transformation. So among all these methods, okay, we're going to be discussing 4, and probably you can differentiate them by the blue font, okay, of these 4 methods, which are retroviral infection, microinjection, nuclear transplantation, and embryonic stem cell transfer. So I'm going to be discussing them in no chronological order. So the first one is microinjection. If you remember, in the first week, just before MCO, in the history of transgenic animal, I did put in microinjection as the first method to be discovered. So it is the first method, a very old method. It has been improved over a period of time, and still the preferred method, and sometimes combined with other method to transfer gene into animals for producing transgenic animal. So in this method, first and foremost, okay, eggs are to be harvested from superovulated animals and fertilized in vitro in the lab. So the keyword is superovulated, which means that the animals are given or supplemented with growth hormone to make sure that they are ready to produce the eggs. So the eggs are then fertilized with a sperm, okay, to make sure that they produce fertilized eggs containing male and female pronucleus, and then being used to whereby your transgene could be injected, okay, into them using a micromanipulator. So in the next slide, okay, this is a picture of the micromanipulator with on your left the pipette to hold the egg, and on your right, okay, a very fine micropipette. Okay, seems like it's not that fine, but when you see it with your nickel, it's actually very fine. This is to basically release your transgene or your transgene in your egg, okay, into there. So this is your egg, okay, with the female and the male pronucleus. So probably, okay, if you were to look at it carefully, this is the female pronucleus, which is a bit smaller. This is the male pronucleus. So you are to inject, okay, your DNA into either one of them. So with this method, all the pipettes, okay, are used for the purpose, and they are custom made, okay, from thin wall capillary tubes. So they are actually very fragile. So with the holding and the inject pipette, okay, it is basically to make sure that, okay, it holds your egg. There is a vacuum, okay, that suck it, okay, while holding, and on the right, okay, is actually to release, okay, the transgene. And this movement is regulated by micromanipulator. Kind of similar if you were to play a game, okay, on PC using joystick. Okay, similar. So you don't really need strong hands, you just need to have a very good hands to control all this because it has to be controlled in 3 dimensions. But again, there is a restriction to this technique, is that talc-coated glove should be avoided. This is why, because we don't want it to be coming out of your hands from your glove, okay, and basically probably get into your culture and contaminating it. And the accuracy of this method is actually could not be asserted. The only more or less indication that the DNA has been inserted and it has been a successful injection is via the swelling, okay, of the pronucleus. So once completed, then the embryo could be transferred to another dish for incubation, and of course, you could visualize, okay, to see whether more swelling has occurred and whether it will be a successful injection or not. So usually in real class, I would show this video by jove.com on these processes, but probably if you're interested, okay, you can just type it out, okay, and search by Google for yourself. Now, some of these valuable embryos, okay, are then transferred to recipient female, provided that there are swelling, but swelling doesn't mean that, okay, the transgene will express. It just means that successful injection, okay. And so far, like I said early on, okay, it is one of the main methods to create a transgenic animal. And with this method, 100 copies of foreign gene, okay, could be injected before implantation to the surrogate mother. However, you have to also bear in mind, not all animals born, okay, using this method or any other method that we're going to see in a while will be transgenic. And so far, only a proportion, okay, of those can express the gene well. So you need to ascertain, you need to do some analysis, and you need to do some breeding. And for successful transgenic animals, okay, the first generation, they are referred to as founder animal. Right. So now with this technique, okay, another restriction, okay, is that you need to do your engineering, you need to do your manipulation before you add the gene into the egg. This is because, okay, with this microinjection method, genes can only be added. Nothing, no modification could be done afterwards. So you need to do your modification beforehand. So the success of this technique relies on careful collection of embryo from productive female donors. Secondly, mastering the microinjection and transfer of suitable female recipient. So those who likes to play video games, might be this would be suitable for you. And the last one, construction and preparation of transgene to be injected, very crucial. So this method is applicable to a wide variety of animals, not only to mice, okay, but to mammals. Second method I'm going to discuss is on embryonic stem cell transfer. So embryonic stem cell and somatic stem cells, okay, they are different, okay, in terms that embryonic stem cells are pluripotent. Pluripotent, okay, means that cells could differentiate and become any cell types. So examples of embryonic stem cells are blood and organ, whereby it could be developed, okay, to produce, okay, any other cell types. So as compared to stem cell, okay, or somatic stem cells, okay, they are undifferentiated cells, okay, they will remain as it is. So this method, embryonic stem cell transfer, is less random than other method because it actually is very specific. Okay? Used usually to target gene sequences to specific sites in the genome and used mostly for knockout. So generally, okay, in this method, stem cells from the blastocyst stage of developing embryos are selected and grown in culture. Target non-essential gene for DNA integration into animal genome, and again, this is the workflow of how it is done, the embryonic stem cell transfer. So you need, okay, to harvest, okay, eggs and make sure that you fertilize it, okay, and then again generate your stem cells in culture, okay, put in your transgene, transfer it, okay, into the blastocyst, okay, and then the blastocyst is implanted into the foster mother. Alright, so for the manipulated embryos, okay, it must be transferred into a suitable reproductive tract, okay, and not just any part, or you cannot just basically deliver it in vivo and whatnot. So recipient females for this method, okay, is recommended to be earlier in its reproductive cycle than embryo donor to facilitate in the uptake or to facilitate, okay, in the pregnancy. The embryos are then transferred into the infundibulum of the oviduct in mice, and of course eventually move to the oviduct, okay, through incision, through surgery. And this infundibulum is actually very similar to a human fallopian tube. I have the picture in the next slide. So after about 2 weeks, pregnancy is expected to be visible. You can actually visualize on the stomach, and this stage is referred to as post-ET embryonic transfer, not extraterritorial. So expected delivery of the litter is about 3 weeks after post-ET. So you have to do your calculation. So this is the diagram or the picture of the mouse ovary plus the infundibulum. Very similar to our fallopian tube. So you have to incise here, to cut up here and put in the fertilized egg with your transgene stem cells and transgene here, and then sew it back, and hopefully that pregnancy takes place. Right. So analysis of transgene may be done at 6 weeks of post-ET, so after giving birth. But certain transgene might be activated in the utero and might affect survival of the embryo or the length of the gestation. Most of the time, it will make it longer than normal. So, to make sure that there is no problem with it, so it would be recommended for researcher to prepare the transgene by homologous recombination and insert it into the embryonic stem cell. So, with this method, homologous recombination, embryonic stem cells mediated gene transfer is preferred for gene activation, especially for the knockout method, because you want to know whether the gene could behave, or whether the systems as a whole, the animal or even us human, could function when the gene is being knocked out. So this kind of method is important for the study of genetic control or development, as it allows precise targeting of defined mutation in the gene, and you can play around with it, and especially if there is a disease involved. And embryonic stem cells have actually been discovered for a long time in mice, but in other animals not so. In humans, it has only been discovered in 1998 and explored since then. So the third method is on retroviral vectors. So retroviral vectors, as you can guess by the names, is using a virus by the name of retrovirus. So it is being used as a vector. So we know that some viruses can be used as carrier. There are lots of viruses. In chapter 5, we're going to be discussing 5 of them. So viral genomes comprise of both genes and cis-acting gene regulatory sequences. So you can read out about it. I'm not going to tell you here the differences between cis and trans-acting. So after separation, the transgene will be linked to the cis-acting sequences and introduced into embryonic cells, taking into account or using the virus's infection method. So location of most of these cis-acting sequences is actually in the terminal regions to enable simple and effective retroviral design, and of course efficient delivery and packaging. So transfer technique for using retrovirus vector is similar to microinjection. So I'm not going to go into details of retroviral vectors because this is actually just kind of like a vehicle, kind of tool to be used. The steps is more or less similar to the 2 previous methods. So different embryos using this method could be targeted because they have different location of genes. Hence if you were to put it at different location, it will result in differences in the offspring in terms of the phenotype and even the genotype. So of course with every other method as well, further selection is required. So the last method that I'm going to discuss is on nuclear transfer. So for those of you who don't remember, so this is more or less the structure of a nucleus, whereby you have nuclear envelope encasing it with nuclear pores on the outside, and you have your nucleolus the center containing your DNA, your genetic material, plus being enclosed or basically surrounded by your condensed chromatin. So with this method, it enables scientists to create an animal genetically identical to an existing animal. So this is the method used to create Dolly. We will see it in a while. This method may be used to create a herd of genetically identical transgenic animal without breeding. May also be used as an efficient method to create transgenic animals, regardless of whether it's a small animal or mammals. For this method, of course, cloning technique is being used to produce those transgenic animals. So the donor nucleus have to come from cell of animals to be cloned, not just any animals, not just any donor. It has to come from the animal that you want to clone because it will have the same property, same genetic makeup. So the basic techniques of this nuclear transfer is to remove nucleus from unfertilized egg and replace it with a donor nucleus, and use electric current for fusion for gene division, and then later on implant the reconstructed egg into the surrogate mother, and hopefully after enough time it will give birth to the founder animal. So the donor cells are obtained either from embryonic cells or somatic cells. It doesn't really matter at this stage, because the special thing about this method is that probably you can see at the last point, is that it could determine the sex. You could actually choose whether you want it to be male or female. So what happens is that after electric current, during cell diffusion, in vitro cell division will occur repeatedly. So you need to make sure that division occurs, mitosis and meiosis occurs before you implant it into the surrogate mother. So like I said, allows predetermination, and more or less this technique seems very easy, but a lot of care needs to be done, because when you remove the nucleus from the unfertilized egg, you need to make sure that you remove totally the nucleus, because if not, then it would contain some of the genetic makeup from the original unfertilized egg. So I'm going to stop here and move on to the next video.